I'm Mark DeBard. I'm the new chair of the Emeritus Academy this year. And you too can serve in this capacity. Just volunteer for the steering committee and volunteer to be an officer, and you'll be in that rotation. It's not a problem. We always need volunteers. We have about 6 meetings a year. Our first one is an orientation meeting on September 12th. The meetings after that, about 5 of them during the year, are 2 or 3 hours each through the year to make this organization run. And we always need people, so feel free to volunteer and you'll likely be chosen. I want to remind you of some deadlines coming up. Friday, September 13th, tell all your friends and neighbors, is the deadline for membership applications for the fall. There'll be another open season in the spring. November 1st is the small grant application deadline, and November 15th is the newsletter submission deadline. I think our dean is doing that this year. So our dean back there is the person going to be doing the newsletter this year. You send her or Jackie, our staff liaison person, the information, and they'll be happy to work with you to get it published. In terms of the steering committee, if there are any members of the steering committee in here right now, would you please raise your hand so we can see you? These are the people volunteering to lead this organization. Volunteer-run organization with a staff person under the Office of Academic Affairs is how we operate. We do have a budget. Most of that budget goes to small grants. The rest of it goes to lectures like this and meeting expenses. Do not forget that next month, the lecture, which is normally on the first Wednesday of every month at 4 p.m. in October alone, is on Tuesday, October 1st at 4 p.m., not Wednesday, October 2nd. This is to avoid a religious holiday that the university celebrates. So it'll be on Tuesday next month. So our talk today is going to be given by Dr. Avner Friedman, who is the Distinguished University Professor and Academy Professor in the Department of Mathematics. Dr. Friedman received his PhD from Hebrew University in 1956. He taught at Kansas University, Indiana, UC Berkeley, Minnesota, Stanford. In 1962, he went to Northwestern, served as the Noyes Professor of Math until 1995, then as the Duncan Distinguished Professor of Math at Purdue until 1987. Then he returned to Minnesota as the director of the Institute for Math and Applications. In 2001, he came to OSU, where he established the Mathematical Biosciences Institute and was its founding director until 2008, becoming a distinguished university professor in 2007. His research interests include partial differential equations and mathematical biology. He's published 25 books, over 540 research papers, served on numerous editorial boards. Since 1987, he's been a fellow of the National Academy of Arts and Sciences, and since 1993, a member of the National Academy of Sciences. His lecture today is on mathematical biomedicine, examples from osteoporosis, lung cancer, and I think Alzheimer's disease as well. Dr. Friedman.
Thank you very much. Actually, what does mathematics has to do with biomedicine? That's a question that every reasonable person will be asking. So that's what I want to explain today. And so maybe I'll go to the next one. Okay, introduction. So introduction. So in mathematical of biomedicine, the starting point is we consider a question that arises in medicine, and we try a question that maybe can be handled with mathematical methodology. How can we do that? We first look at the biology and the most important factors which would be cells, proteins, DNA, or elements like this and other molecules that are essential in order to state the question that arises in the biomedicine. And then we set up a network that relates how each cell, for example, talks to any other cell, how proteins affect cells, and so on. There is a network. We express this network by mathematical equations. And these equations, like, say, differential equations, and these equations have parameters. We have to find these parameters. We have to look at the literature and try to identify as much as we can, as precisely as we can. Not always we can do this, of course. And then we simulate the model and see what we get. We test it against some maybe experimental results in mice models or clinical trials and so on. And we feel that now the model makes sense, and then we address the question by simulations. And I'm going to give you 3 examples. And I chose examples that are from recent work, but also that are completely different topics in medicine, just to show you the variety of problems that may arise. So the first one is on metastasis, okay? So I chose metastasis into the bone, because that's a really problem that can be mathematically handled very well. And I chose also breast cancer, because breast cancer produces a certain kind of microRNAs that are that are related to bone metastasis. Okay, so now, first of all, what is the bone like? The bone is a dynamic organ. It is a source, our source of calcium. And the outer part of surface of the bone is continuously resorbing in order to provide calcium through the brain to the cells. So there are cells that are resorbing the surface of the bone, and these are the osteoclasts. Let's see, oops. These guys, where are they? Here they are. See, they are breaking the bone. Also here, these guys here, they are breaking the bone. But then, or here, they are breaking the bone. But then there are bones that, there are cells that producing bones. These are the osteoblasts. So osteoclast breaking it, resorbing it. Osteoblast forming the bone. And they work at each point on the surface of the bone together. One resorbs, the other comes and fill it in. So that's basically what, the bone is a dynamic thing. And there has to be equilibrium between the 2 processes of resorbing and breaking. Okay. So now, going back to breast cancer. So they have to, when a cancer cell separates from the primary cancer cell and want to go to metastasize, it has to choose a place where it can really colonize, what's called the fertile soil over there. And what cells do, okay. So, but first of all, the question. So what is the question that we ask in this problem? How does the cancer cell would like to go inside the bone, because there's a bone marrow, it's a large, very rich, very good potential for metastasis, but it has to break the bone surface. How does it do it? That's a question that we consider here. So, the osteoclast I mentioned, osteoblast, and so, and again, in normal situation, there is a normal brain bone remodeling, and I want to show you how it is, okay? So on this, the right arrows here are the process of bone formation. These are, it starts with mesenchymal stem cells here that differentiate into osteoblast precursor or precursor osteoblast, and then mature further to become osteoblast, which form the bone. They produce things that form the bone. On the other column here, there are myeloid precursors cells, and they are differentiate into osteoclast precursor, which then go into osteoclast and bone resorption. Forget about this red things in red squares. Okay. So now, how does the modeling happen? The normal modeling happens. The osteoblast is producing a ligand called rankel. The osteoblast here, precursor, have a receptor, rank, and when they combine on the surface of the osteoclast precursor, it differentiates into osteoclast cell and goes here. This is how the forming inform on the osteoclast that is going to resolve. So that's one way. On the other hand, when bone is resorbed, there is a release of TGF-beta here, which activate the mesenchymal stem cells and tell them we need to form a bone because we just had resorption. But this balance has to be controlled if it has to be regular in a normal situation. What controls it is little things here, OPG. This is also a receptor on osteoblast. There are too many of the rankel, there will be too much of this arrow here to us, and there will be too much resorption. And if there is too little, okay, if there's too much. So what happens is this OPG combines with RL and remove some of them from being active. It blocks them. And therefore, this process here slows down. This is a blockage here that you see. So this is the normal bone remodeling. This is a rough picture. It's rough enough, but in order to explain what you want to answer the question, how, okay. So I already explained what this rankel thing does and what the OPG, and OPG is a modulator, modulates the resorption and make sure that the modeling is done the correct way. Okay, exosomes. Okay, these are vesicles, very small, 30 to 100 nanometer. The size of a cell is about 10 micrometer. So the cell has a lot of vesicles, and cells are secreting these exosomes. The exosome contains proteins, microRNA here, RNA, fragments of DNA, lots of stuff. And this is like the cells secretes thousands of them just into the blood to find out what is the neighborhood like. This is like scouts. And this is a very active area of mostly very technologically difficult to identify what in the blood, some microRNA, for example, where it is coming from. And the reason I chose breast cancer, because we know about microRNA that breast cancer produce. So we have this information, okay? So what this breast cancer try to do is to weaken the bone ahead of time, because once a cancer cell comes in unprepared near the bone, it has to wait several days or maybe several weeks until the bone is broken so it can get in. Okay, so we know, for example, the microRNA 21 and microRNA 2014 are produced by breast cancer, and they are degrading. Here they are. They are activating from the production of osteoclast. So is 214 at different levels. And therefore, by producing them, the cancer cell ahead of time tries to resorb the bone, to break the bone, to decrease the density. So here you see, this is a equilibrium remodeling that I mentioned, and you can see here what they do. They do other bad stuff, but here, let me focus on this 21 and 214. I have to have at least one formula in this talk. So here is a formula. It shows you how the osteoclasts are created by, this is something that controlling OPG appears here. And this is going from precursor to osteoclast, but you see that microRNA 21 increase this production. So there are more osteoclasts than there should be, and therefore more bone resorption. Bone starts to break. Before the cancer cells are moving in, preparing the bone for metastasis. Now, what can we do about it is not clear. Oh, these are some more equations. Okay, here is a bone forming by osteoblast, bone resorption. Oh, there is I just put this, one of the microRNA that the breast cancer produces, and the result is the bone density is more minus, more resorption. And the bone simply gets so weak that the cancer cells later on, maybe a week later or a month later, we don't know, but they can get into it. What we say that they prepare the bone for metastasis, called pre-metastatic niche. Okay, so that's okay, there's some drugs that can be used to strengthen the bone, but and there's some examples that if you knew person is going to, if I'm a doctor, I know that this patient is going to have bone metastasis, I may give him these drugs. And you see they are, instead of the bone density, this is 1.1. This is in units of gram per centimeter cube, the bone density. instead of going down like that, it will stay in the normal between the two horizontal lines. This is the normal level, but of course we don't know that. There are some recent work trying to say, can we block microRNA 21, for example? So there are research articles about it, but it's very tricky to try to do something like that. Who knows what are the results, what the consequences will be dealing with RNA or microRNA in this way. So this is ongoing work and may eventually lead to something. Okay, next example is osteoporosis, completely different. Now, what is, okay, first of all, let me tell you, again, when I think about it, that it also has to do with the bone, so I don't have to repeat what I thought about bone forming, absorbing. So the bone density growing with age until the age of 30. Then it remains more or less uniform until the age 50. Then it starts to go down. Because we are looking at osteoporosis, we are going to look at people age beginning 50. And this is a standard tables, graphs that tells us how the density is decreasing from over the years, you see, from age 50 to age 100. The top one is for men, and the second one is for women. This is the sharp decline here is osteoporosis period. I'm sorry, is a menopause period. A sharp decrease in there. So this is a graph that's for men. And in our model, we are trying, when we try to verify that the model is okay, we are going to compare, we find out by simulations that our graphs here are close enough to these graphs. So, oh, okay. Yeah, thank you. All right, so where are we? Osteoporosis. Okay, so how do you define osteoporosis? Basically, the disease is such that if you fall, there is a danger that you are going to break a bone. And it was defined to be a state where you have lost 25% of the density you had at the age of 50, 25%. Okay? Okay, now there is again a problem with the bone, and this is again the bone remodeling going to be broken by a completely different mechanism. The question is, how do you characterize aging? What happens in aging? What are the signals of aging? So one of the primary markers of aging is the appearance of cells, same as in mesenchymal stem cell, for example, that are senescent. Senescent. What does senescence mean? A cell is in a state of senescence where it's still alive, metabolism is going very well, but it cannot reproduce again. It had divided many times, and finally it stops dividing. So it just stands there. It doesn't die, but when you just stand there with nothing to do, you run into trouble. So what the senescence they do, they produce various molecules that are mostly have a negative consequences, negative consequences. So, first of all, they are inflammatory, and also, so I'll show you what they do. So okay, here, the inflammatory. You see the IL-6, interleukin-6, is produced by the senescent cells. They also produce microRNA, like 34, that interfere with this process of control. You see what they do? Here they decrease the the OPG, and therefore there is more RNkel ready to do this and more resorption. Also, the mesenchymal stem cell, instead of all of them going towards construction, towards forming of a bone, some of them by this microRNA, 153, they are going into adipocytes, which doesn't do anything for the just fat cells. So in this way, as people get older, there is increased disruption of the bone modeling tending towards resorption, and the bone weakens, the density goes down. So that's what the model says. Well, this is just how mesenchymal stem cells are introduced. You see, they start here, they increase, this is times this factor. This is T times, we start at the age of 50 and start with 0 mesenchymal cells, and then it starts going on and for 50 years. This chi is, you know, the biological age is different than the chronological age. This chi, if it gets larger, if it gets smaller, then more mesenchymal cells are coming to the normal thing. Normally, chi will be 1, but more senescence when people age, when chi decreases, this is a person whose senescent cell accumulate faster. Okay, now here is okay, so what do? We have to, I mean, if we couldn't do anything with the metastasis into the brain to stop it. For metastasis, you give chemotherapy, and that's it. But here we can do something, because there are drugs that eliminate senescent cells. So this is ongoing research the last few years. There are various different types of drugs. Some of them are just taken from some vegetations. And one of them that I use here is fisetin. So fisetin is one, quercetin is another one. So fisetin. And now if you give this drug, so it eliminates senescent cells and therefore relieves or slow down the progression to osteoporosis. So, so here we show a graph that when it's given at a certain time, the bone density will be elevated. If you don't give anything, it will go like the blue line. If you give it, you are going to gain something. And maybe I can show you better with some tables. okay? Suppose you take the drug. So I assume that, so this is for women. So I assume that estrogen treatment was given and followed with later on with fisetin that eliminates senescent cells. So if a person takes a drug at the age, so this is a, oops. Okay, this is the initiation when you start taking it, and this is the end time. So if you take the drug at the age 60, by that time you lost 10% of the blood, of the bone density. Say at the age of 80, you would have lost 26% of the density that you had at age 50, 26%, which is like almost like what they define as osteoporosis. But if you did not take it until at the age of 70 or 75, the bone density loss will be larger, 28% or 29% if you start at 75, at the age of 75 to take it. You see, 29%. So altogether, what it does, what this says is, this table, it says that the earlier you take fisetin, and you take it all the time, I don't think it has any serious side effects. So if you take it, starting it early, at the age of 60, you are going to gain by decreasing the loss of bone density, not significantly, not very significantly, but somewhat. So it's a modest kind of thing, but the drug doesn't have much side effects. So, you know, take it like maybe like a vitamin or something. So that's what the table shows. Okay. So now I come to Alzheimer. So this is one of my postdoc. She's from Korea. And okay, so the numbers are large, and they're going to get larger. And the expense is huge, but of course the pain the family suffers with the person that has the disease cannot be measured by money. And it has no cure. No cure. We don't know the reason. We don't know what's the cause of it. If we knew the cause, then we maybe can do something. But we know we have some things recognizable that are associated with Alzheimer. This is beta amyloid or amyloid plaques. The amyloid plaques are made up of amyloid beta peptides. So that's one thing. Second thing is there is tau proteins in every cell, in particular in neurons. These tau proteins, they have a job, or maybe one important job, but some other jobs, to create microtubules in the cell so that stuff that is being made at the nucleus can move around and go to other places all over the cell, the tau proteins. But in Alzheimer, there are too many of them. They are hyperphosphorylated. And instead of building these microtubules, they get all messed up into tangles, neurofibrillatory tangles, and that actually damages the cell. So this this hyperphosphorylated tau protein damages the cell. Beta amyloid. So beta amyloid is like this. You know, there is on every cell, but particularly in neurons, there are surface proteins. They're called APP. They are amyloid precursor protein, APP, amyloid precursor protein. And these guys have a role in the cell, but somehow they break down in beta amyloid inside the cell and beta amyloid outside the cell. Outside the cell is what we see later on in the plaque. And they are doing it, and the question is in abnormal way. We get too many beta amyloid outside. So every normal person, healthy, not normal in the brain sense, but in the body, in the biologically normal, has amyloid beta, and that can be measured in the brain. But they are more so in the case of Alzheimer. So they see it in PET scan, they see this amyloid, beta amyloid plaques, and they see these tangles of neurofibrillatory tangles. So let's say NFT, let's call it NFT. So this is recognition. This is something associated with Alzheimer, but we don't know the cause. So what we want to do is to understand, you see, okay, here's a question. How do you define in mathematical terms the concept of this guy has Alzheimer and the other guy doesn't? We cannot say, well, cognition is not very well. It cannot. The person cannot lift a glass. Well, it cannot repeat 4 words after another. You know, I forget. If you give me 4 words, you cannot repeat them in order. And things. We cannot do this in a model. But fortunately, we have a mathematical model for Alzheimer. Or sort of. And here is what, here it is. You see, the concentration of amyloid beta was measured in health and measured in Alzheimer, and it was found that the average in health is 1,000 nanogram per gram, whereas in Alzheimer, it is 6,700, 6 or 7 times more, the average. The tau protein, the average number is 137, it's much smaller, picogram per milliliter. And in Alzheimer, it's 490, about 4 times more. Now here, you see the interval, the range in the normal is at most 300. That's where the Alzheimer patient starts. Over here, it's from 50 to 3, it varies from person to person, of course, to 3,500, and this only starts here. So there is some overlap here. But we take to say that this number, 6,700, and around it, that will be for us an Alzheimer case with some uncertainty. And again, 137, 490 will be the sign for Alzheimer. Okay, so now, let's see, am I here? Yeah. So now, what I want to, so again, we don't know what the primary what causes all of that. But it is seen, it is observed, the inflammation in the brain, in the neurons, is a very early event, very early event. So we simply say, we have inflammation here. You see R of, depending on time, X and Y in the plane, in the brain, on the surface, some of the layers in the brain. We have a time T, we have this amount of inflammation, which affects the beta amyloid. They make this APP that I mentioned divide faster. It creates more beta amyloid. We have the normal. We have R with the bar here, the same inflammation, but it affects the hyperphosphorylation of tau proteins. Two different profiles, one for beta amyloid and the other is for tau proteins. So this is our assumption. And now, and this is a model, this is a network here. So network has here, well, here is a neurons, which has inside the internal beta amyloid, there is a tau proteins. The tau proteins creates these tangles. And when the cell here produces an external beta amyloid, this is the astrocytes, which is the cells that are brain cells that are all over, and they are protecting around, they are hugging the neurons. And this is microglia, which is like macrophages, taking care of clearing up things. So that's a basic thing, the minimalistic model. There are other variables involved, but we are not including them because we want to focus on the PET scans. And I'll tell you later why. Okay, so that's, so here is how the inflammation increases. There is a basic inflammation here initially, and it starts to go, here is T over a constant. So let's say like T, go like T, and constant plus T. So it goes in a modulated way. It's a curve which is, I cannot draw it here, but it's going, started going sharp and then stabilizes more or less. So now we want to take from inflammation to what we see in inflammation to what we see in the actual profile of the beta amyloid and the profiles of the tau tangles. Okay, so here are various equations which is based on this model, on this picture that I said that of the networks. And now here's what we simulate. We assume, we take this to be, this is the map of pixels of different levels of inflammation, larger, smaller, and so on. We simulate the model for 500 days, and we find out that the amyloid beta, because of this inflammation, has this profile. And the density of the amyloid, or beta amyloid, varies from, the blue one is 4.5, this is 6 times 10 to the minus 6 gram per centimeter cube. Different inflammation is for the tau, it causes hyperphosphorylation, and it also leads to this map. So what we say is, with this inflammation, we have a passage from early event of inflammation to what we should see after in the brain. Because that's what we see should be the picture in the brain. Now, to be a little bit more concrete, here is the brain. There is inflammation spread around. That's what you see here, beta amyloid, and that's what you see with the tau proteins. This is just one. On the left side, the 4 figures is for one patient. On the right side is for another patient. I tell you why I gave you 2 different patients. Why I had the patients to give you 2 different patients. There is a theory that says beta amyloid is a source of all troubles. In fact, there were so many clinical trials that tried to eliminate beta amyloid and by that solving the, addressing the disease. And finally, there is 1 or 2, now 2 of them, that partially, very partially delay the symptomatic growth of the disease. It's just delayed. We cannot stop it, but delay it a little bit. So they say beta amyloid is a trouble. Let's aim at this. There is a minority. I just judge by the number of papers that says, no, tau protein comes first. And once it develops, it also causes beta amyloid. But that's tau protein is more primary than. What I'm saying is that you are right and you are right. And how can they both be right? Because none of them really is complete. They are countering. This is called the first one is called the amyloid theory. The other one is called the tau protein theory. And each theory can explain things, but not everything. So we can't really accept each of them as such. But if you assume that everything starts from inflammation, then it just depends on the inflammation, how it occurs. You see here on the left side, you see just by the colors that beta amyloid is much stronger than tau protein. This belongs to amyloid theory. Here you see we took inflammation such that this is more extensive than here. This would be the tau theory. So it's not a matter of this or that. It depends on how the inflammation began. Okay, now what's that? Now here's the thing. Alzheimer is a disease that takes time to develop. It is believed that it takes at least 10 years, maybe even 15 years. And the question is that we are asking, take this drug that eliminates amyloid beta. Suppose you gave it not when symptoms began to show up, but earlier. Would this delay the disease, the onset of the symptomatic disease later? Will it postpone it later? So there is a lot. There is several papers now dealing with what is called longitudinal research into Alzheimer. They're going back and say, during these 10 years, could we have some sign that this guy is going to develop Alzheimer and then do something for this guy? Of course, they don't do it with people. They are trying it with animals. And you have to have an animal model of Alzheimer. And so we asked here the same thing. Suppose we knew what will happen if you give the same drug we are using today that delays, that destroys beta amyloid and postpone the disease by, especially if it's given when the first signs of cognition problems show up. What happens if you give the same drug 200 days earlier? So here is what we are producing with some inflammation. We look at the PET scan. We just did it with squares, but it could do it with a human brain, the size of human brain. So here is every 100 days keeps going up and up, more and more red, more and more beta amyloid, more and more tau proteins. And at some point here or here, it is reached the level where we say this is an Alzheimer case. We are approaching Alzheimer. We already passed Alzheimer. We cannot say exactly one, but so we try to simulate and see this is the density, just the density of the beta amyloid as it grows, the average density, and this is of tau protein. Now, this is the important thing here. The horizontal dotted line is the level of beta amyloid by which we say this is the average case of Alzheimer. The beta amyloid density keeps growing, and it is not yet, you don't have the disease yet, you don't have it yet, you don't have it yet. You reach this point, and you surpassed it. Now you are in the Alzheimer zone, and maybe even in the symptomatic level. If you gave this drug that blocks amyloid beta earlier, at day 2000, so here I took it from 15, that's about 10 years here, horizontal axis. If you gave it at day 2000, the green spot here, you will reach the level of Alzheimer here at the day, say 3000. So in other words, the symptoms will appear 1000 days later. You have gained 1000, you have gained 3 years postponement. Of course, we don't know that the person is going to develop Alzheimer. I mean, everyone has beta amyloid growing, but we don't know which one is going to be. But theoretically, if we could do that, we are going to gain this. This is for beta amyloid, and this is similarly for tau proteins. So, but in any way, this is sort of conceptual way that will go along with longitudinal studies. As more and more information is available, we will get better idea about the dynamics of these curves. So that's a conclusion on the Alzheimer's disease. The question will be, when will be the optimal time to start? If you start too early, then you are not sure that it's going to be a case of Alzheimer's. If you start too later, you don't gain much benefits. So that's will be the dilemma, and that's where I like to stop. Thank you.
Jackie's going to come around with the microphone, and so am I, apparently. For anybody who wants to ask questions of Dr. Friedman, right? Who's first? Can you do that, Jackie, back there? Yeah. Got a microphone.
So if I understood correctly, in the first two cases, you solved a model that is a coupled set of ordinary differential equations in time. And my question is, can you say a few words about how you validate this model before you make these calculations that you showed us?
So I think you said that was expert consensus is the way you figured that out. Expert consensus as opposed to total validation. Yeah. Other questions? Yep. Carl?
Two questions are,
Oh, okay.
what animal model did you use? That's the one. And then the other one is, what about anti-inflammatories? If inflammation is the culprit behind the amyloid and the deposition and the tau, what about anti-inflammatory drugs?
You mean to use anti-inflammatory for, in any case, in any of these diseases? Yes, I mean, this would be a drug that is I don't think that, I mean, because we don't know that Alzheimer begins with inflammation. We don't know that. We assume, because it's one of the primary things that they see right away when you see someone has Alzheimer, immediately you see that inflammation. We think that, so we are basing it on the model in Alzheimer on early inflammation. But if we knew, here is a patient that yesterday was not did not have any inflammation, today he has inflammation, and later on it becomes, and this is going to be inflammation that causes Alzheimer, we'll immediately use a drug to block the inflammation. But whenever possible, of course, we use anti-inflammatory. In cancer, we use it all the time. You know, I mean, giving, I don't know, prednisone or things like that.
Dr. Friedman, the other half of his question was, what's the animal model you're using?
I'm sorry?
What is the animal model of Alzheimer's that you're using?
Animal models?
The animal model, you mentioned an animal model?
Well, the animal model in Alzheimer, you know, last year there was actually a guy here, a speaker, that talked about Alzheimer, about neurological diseases with various production in the brain of various chemicals. I mean, I don't know how they make them, but they have to make a mouse that you go through labyrinth, for example, and several times, and then a minute later it forgets how to get out of it.
Okay.
So you gave this mouse a certain kind of drug. You have to give it you have to give the animal a drug that will, so that the effect will be that it's just confused.
Other questions? All right, sir. Thank you very much for your time.
Yours.